talking about an important subject, that's infectious diseases. And we are going to have a nice round table, I believe. We start with Professor Miguel Prudencia, that all of you know. He's going to talk about vaccination. Then Professor Luís Graça is going to talk about antibody production. Professor Thomas Enscheid is going to talk about methods of diagnosis. And I'm going to talk a tiny bit about new treatments. And we are on time. Professor Miguel Prudencia, please do.
Okay, so good morning. Thank you very much for this opportunity and to the organizers for having me here today. I'm going to talk about, so the four of us are going to talk about different aspects of infection, from prevention to diagnosis to treatment. And so I am starting with actually the prevention issue, and prevention against infectious diseases largely, although not solely, but largely means vaccination. And so the talk that I'm going to give now concerns disease prevention through vaccination. And what I'm going to do is a bit of an overview of the importance of vaccines in general, and then a little bit at the end about our own work at the Instituto de Medicina Molecular on the development of a vaccine against malaria. And so let me start with telling everyone, for those of you that don't know, what a vaccine is. And a vaccine is essentially a biological product that teaches our immune system to recognize an infectious agent as a foreign entity, to remember it, and this is important because vaccines only work because we have memory of that pathogen that we recognize, that we learn to recognize, and then to destroy that agent upon a subsequent encounter. And this is what is aimed at with vaccines, is to confer long-lasting protection against an infectious microorganism. And of course, this is done through a complex array of immune responses that our immune system is able to mount, and which in very simplistic terms, but I think useful but simplistic, it's our adaptive immunity that makes this possible through the production of, on the one hand, antibodies, and on the other hand, T cells, both of which play complementary roles in the protection against the infectious agent that we are vaccinating against. And so it is very hard to overestimate the importance of vaccines throughout mankind's history, or at least since they exist. And I would say that vaccines are certainly among the medical achievements that have saved the most lives since medicine exists. And I've tried to find numbers about the estimations of how many lives have been saved by vaccines, and there's controversial numbers about this, but this very recent paper published this year tells us that this estimation is certainly in the tens of millions of lives that have been saved and will continue to be saved by vaccines in the world. And as examples, this is the drop in the cases of three very prevalent diseases, mumps, measles, and rubella, since the introduction of vaccines. And if we look at this chart here, which I think it's probably too small for you to see, but essentially what we have here is the decrease in the number of cases and the number of deaths attributed to an array of infectious diseases, and the decreases in cases and deaths that have been possible due to vaccines. This is in the US, And it's also the case in Portugal and in Europe in general, but unfortunately, this is not the case in all the world. In our so-called Western world, we have learned to forget about some, or almost forget about some diseases because we have vaccines, and we almost forget that these diseases not long ago would be taking the lives of millions of people, and that we no longer worry about these diseases because vaccines are there to protect us against them. Unfortunately, like I said, the coverage, the vaccine coverage is not exactly similar for all diseases and all around the world, and so we still have many parts of the world where vaccination is below what would be desirable, and for some diseases we have much better coverages than against other diseases, and it's important to make sure that mechanisms are in place so that vaccines can reach everybody who needs them. So what does a vaccine, how does the vaccine do what we expect it to do? So how do we teach the immune system how to identify the organism against which we are vaccinating? Well, there are several ways to do this, and traditionally or historically, one of these ways is to use the infectious agent itself in a live attenuated form that will be presented to the immune system, who will then learn to recognize that agent and mount a response against it. This can also be achieved with inactivated agents like inactivated bacteria or viruses, and we have multiple examples of vaccines against bacterial and viral diseases using this strategy. Or, in the case of diphtheria and tetanus, what is used is an inactivated form of a toxin that is produced by these bacteria, and this is used as the vaccine and is used as the way to teach the immune system to fight the disease caused by these bacteria. And finally, we have subunit vaccines, so vaccines that are based not on the organism as a whole, not on a substance produced by the organism, but on a portion, a fragment, a subunit of that organism that is representative of that infectious organism, and in that way, our immune system learns that by mounting a response against that fragment, it is mounting a response against the organism that harbors that fragment. And this has been, those four options have been the main options for vaccine production until recently. But then, as you all know, in November 2019, a new disease struck, COVID-19 struck, and 2 years later, it had caused the deaths of 5 million people, an estimated 5 million deaths caused by COVID. And of course, we all know the impact that this has had in our world, and it's been obviously a very negative impact on the health, and on the economy, and on society as a whole. But it has had one positive aspect that comes with it, which is the development of a whole new class of vaccines that are the nucleic acid vaccines. And here, instead of administering the organism as a whole, or administering a protein of that organism, of the virus, as a way to stimulate our immune system, what these vaccines do is to give our organism the instructions, the genetic instructions, the genetic code that enables our own cells to produce the antigen, the protein of the virus that we want to immunize against, and then it's our own cells that make this protein, which is then presented to the immune system, and the immune system mounts a very efficacious response against that protein, which is then a response that can combat the virus as a whole and combat infection as a whole. This has been a major revolution. It's made the cover of Time, for example. These are two researchers from BioNTech who pioneered this technology, and the world of vaccines is probably never going to be the same again, because these nucleic acid vaccines offer immense opportunities for future developments of other vaccines, therapies, etc. But so, talking about vaccines against COVID, if we look at the current situation, there are dozens of vaccines in different stages of clinical development, in different phases of clinical evaluation, and these include examples from all those categories that I listed before. So inactivated vaccines, weakened vaccines, we have subunit vaccines, we have, of course, the nucleic acid vaccines. And in Portugal, as in Europe, we've had 4 vaccines that have been authorized for COVID, two viral vector vaccines by AstraZeneca and Johnson & Johnson, and 2 RNA vaccines by BioNTech, Pfizer, and by Moderna. And with this, we have been able to vaccinate a large proportion of the population. Regardless of the type of vaccine that we are using, the process is that we are presenting to antigen-presenting cells the spike protein, which is the protein of the virus that is used in these vaccines. And then there is a sequence of events that will lead, on the one hand, to the production of B cells that will produce antibodies that can block invasion, block infection by the virus, and on the other hand, cytotoxic T cells that are able to destroy the cells that have been infected by the virus and play a pivotal role in protection against severe disease. And so, with this, very recently, only actually a couple of days ago, the European Centre for Disease Prevention and Control has issued an estimate according to which nearly 500,000 lives have already been saved by COVID-19 vaccines, and this is in less than 1 year. As you know, the first vaccine against COVID was administered in Portugal on the 27th of December 1970. You can ask how I know that. I know that because it's also my birthday. And since then, it's been less than 1 year, and more than 500,000 lives are estimated to have been saved with these vaccines. And so, vaccines save lives. Vaccines have been saving lives for years, for decades, and vaccines will continue to save lives. But there are still diseases, infectious diseases, for which an effective vaccine is not available. And it's important that we don't forget that, because these are diseases that are still causing a lot of suffering worldwide. And one of these diseases is malaria. Malaria is a disease that affects mostly sub-Saharan Africa, the sub-Saharan African region, but not only this region. It's estimated to cause more than 400,000 deaths every year, and more than 2/3 of these deaths occur in children under the age of 5 years old. And until very recently, there was no licensed vaccine against malaria. On the 6th of October this year, so a little over 1 month ago, there was a landmark day on the fight against malaria, because the first vaccine against this disease was endorsed by the WHO, and this vaccine is called RTSS. It's a vaccine, a subunit vaccine, based on one specific protein of the Plasmodium parasite that causes malaria, and this protein is called the circumsporozoite protein, and RTSS uses circumsporozoite protein as the antigen to elicit an immune response against the parasite, and therefore a protective response against malaria. Unfortunately, this only affords about 30% reduction of clinical episodes of severe malaria in children, and so it is obviously an important step forward, but it's not the final step forward. We need better vaccines against this disease, which still causes so much suffering, and so this is where other approaches to vaccination against malaria come into play, and the most successful ones have been the whole parasite, the whole organism vaccines based on the parasite itself in an attenuated form. And so far, until recently, these strategies all relied on attenuated forms of Plasmodium falciparum, which is the human infective, the deadliest human infective malaria parasite. And so whole sporozoite or whole organism vaccines based on Plasmodium falciparum have been investigated for quite some years. They show promise, they are very efficacious, but they also have a few drawbacks, and they also have a few limitations, a few challenges. And this is why a few years ago at IMM we have initiated a project towards the development of an alternative whole organism vaccine against malaria based on the concept of cross-species immunity, because there are malaria parasites that infect humans, but there are also malaria parasites that infect other mammalians. And Plasmodium burgii is one such parasite that infects rodents and does not cause disease in humans. And so in a nutshell, what we have proposed is to use Plasmodium burgii as an immunization agent against human malaria by genetically modifying this rodent malaria parasite to insert and make it express antigens of the human malaria parasite. It's like we disguise the rodent malaria parasite of human parasite by placing a coat of human malaria parasite protein on the rodent malaria parasite. And in this way, we expect to combine cross-species immune responses because of the similarities between the two parasites with targeted immune responses that arise because of the presence of a specific antigens of the human infective malaria parasite on this vaccination platform. And so we have made this happen through the engineering of PBVAC, which is a Plasmodium burgii, a rodent malaria parasite that expresses the Plasmodium falciparum circumsporozoite protein. If you remember, it's the protein that is used for RTSS. It's the most potent immunogen that we know in Plasmodium falciparum, the human malaria parasite. And so what we have made is a chimeric rodent parasite that expresses the Plasmodium falciparum, the human circumsporozoite protein at its surface. And in this way, we expect to have cross-species immunity against the human parasite, as well as targeted immune responses that arise from the insertion of this specific antigen on our vaccination platform. And so with this, and this is my last slide, it's my one before last slide. We have spent quite a few years going from preclinical studies to the clinic, and in our first evaluation in the clinic, we demonstrated that this vaccine candidate, PBVAC, is able to afford 95% reduction in liver infection by the human malaria parasite, which is very promising, but is not enough, because the human malaria parasite needs to go through the liver before it reaches the blood and causes disease. And 95% reduction is not enough to completely block the appearance of parasites in the blood. So we are now working towards going the extra mile that is missing, so the 5% that we are missing in order to completely block infection during the liver stage of infection. And for this, we are creating better vaccine candidates that express multiple antigens of different stages of the Plasmodium life cycle, and hoping to create what we call a multi-stage vaccination platform, a multi-stage vaccine against the human malaria parasite. And this is work that is currently going on, and we are hoping and hopeful that we will have the second generation of this Plasmodium burgii-based vaccination strategy in the near future. And with this, I'd like to just thank everyone that has been involved in the work that we carried out over more than 10 years on this vaccine, and of course the funders that made it possible as well. And thank you all for listening. Thank you. So next we have Professor Luís Graça from Faculdade de Medicina, da Universidade de Lisboa, and also group leader at the Instituto de Medicina Molecular, who is an expert on an immunologist and an expert on immune responses, and he's going to tell us about regulation of antibody production. Thanks a lot.
To follow this talk about vaccines that had made my life easier. I translated many slides to Portuguese, so this will look like a movie with subtitles. Anyway, I want to start by saying that the immune system evolved to solve a problem that we have. And this problem is to face a world where we have the ability to mutate and change our genetic components once every 20 years or so. That is our life cycle. But we have to face organisms that have shorter life cycles and that, as a consequence, can accumulate mutations and diversity very quickly. And we know that from experience, that we can become immune to the flu virus from this season, but this will not help us to be protected against the flu virus from next season. So we had to develop an immune system, we mammalians, that can anticipate the changes that microorganisms can evolve for the next season. And this is achieved in a remarkable way. So with a few genes, the immune system can recombine segments of these genes to generate a huge diversity of receptors, and this is changed in the DNA, meaning that each different immune cell has a different receptor, but because the DNA has changed, each cell will only have molecules that are exactly the same. And this is the key for our ability to anticipate the next pandemic and be confident that our immune system can make an immune response against the pathogen that is involved in the next pandemic. This is also a good opportunity to tell the audience something that they do not know. That is the fact that while we think that all our cells have the same DNA, this is true except lymphocytes. Lymphocytes, they change their DNA like this, so each lymphocyte has its own DNA. Now, why this is important? So this was the discovery from Landsteiner. He got the Nobel Prize for discovering the blood groups. It was in Vienna. But the most amazing experiments he has done was to check the immune responses, the antibodies that were produced following immunizations with different proteins. He injected rabbits with a protein, and this rabbit will make an antibody. Another protein, another antibody. And tired of checking rabbits with different proteins, he decided to synthesize proteins that do not exist in nature because of these L and D isomers. And even molecules that do not exist in nature can elicit an antibody. So this led to the concept that it does not matter what will be the mutations from the flu virus from next season, the immune system has the ability to make antibodies against this flu virus. It does not matter what will be the next pandemic, our immune system will have antibodies and cells that can respond to those antigens and make antibodies against those pathogens. Now, the drawback for this is that it is necessary to expand a few cells, the few cells that respond that are specific to this virus, from many cells that are specific to all possible viruses that we may face in the future. And this takes time. So I will skip this in interest of time. Okay, and this takes time. And that is the reason why we have to rely on innate mechanisms for the first few days until the adaptive immune response that is comprised by T cells, cytotoxic cells that kill the infected cells, and B cells that produce antibodies that neutralize the virus. And these will only kick in after a few days. Now, what we can achieve with a second exposure to the antigen is that we can boost with this secondary response is faster, and it has a greater magnitude, leading also to a greater level of antibodies that are already produced. And this is an advantage, for example, for vaccines that have 2 doses, because after the second dose, you have this secondary response, and this secondary response will leave us with a better protection than after the primary response. If we have an exposure, the exposure to the virus will act as one of the 2 responses. So then with just 1 dose, it is like to have a secondary response. Now, what happens, and this is data from a study in Israel and UK, is that although antibodies can decline more quickly, the protection against hospitalization, as you see here, declines over time quite slowly. And here you see that younger people that have a quick decline of antibodies as well, or relative, that have some decline of antibodies, the risk for hospitalization does not change that much over time. And this can be explained from the fact that the antibodies and the protection that is conferred by cellular mechanisms, namely CD4 T cells, CD8 T cells, NK cells, and so on, they may not change as quickly and with the same kinetics as the antibodies. In the same way, if we are facing a new variant, and now everybody is very concerned about the Omicron variant, of course that maybe the antibodies may be less effective against this new variant, but since the T cells do not recognize the molecules that antibodies are directed against, may not be so perturbed by this new variant. And the overall protection, and especially the overall protection against disease and severe disease, may be more or less maintained over time. Okay? Now, when I started immunology, immunology was very simple. That's the reason why I decided to study immunology. And the immune responses that were known at that time were Th1 responses and Th2 responses. Th1 responses were against virus and so on. Th2 responses were against some sorts of parasites like helminths, and they were also involved in allergy. Now, the panorama is more complex, and part of the problem of immunologists is to understand why these different cells are important to drive the immune response in different directions. And we also need to consider regulatory cells that are implicated in regulating this type of immune responses. What we are especially interested in is in how T cells interact with B cells, making B cells make different antibodies. And as I mentioned, Th1 cells and Th2 cells may drive the B cells to make different antibodies, more suited to fight virus or more suited to fight, for example, helminth infections. And it was recently known that not all CD4 T cells can help B cells make antibodies. This is a job that is exclusive for TFH cells that express this CXCR5. If in mice or humans that don't have this CXCR5, they cannot make antibody responses against these pathogens. So TFH cells are critical to make antibodies produced by B cells. And the hypothesis that we have been addressing in recent years in our lab is to investigate if, in the same way that we have Th1 and Th2 cells, we may also have TFH cells specialized in type 1 and type 2 conditions. So we addressed this problem in different ways, and one of the side effects of this was the realization, in collaboration with Helena Florindo from Faculdade de Farmácia, that it is possible to use some nanoparticles that can boost the adjuvant effect of the immunization, leading to greater immune response compared with immunization without these nanoparticles, as it is represented here. But anyway, we are interested in finding type 1 and type 2 responses, so we immunized mice with adjuvants that drive type 1 or type 2 immunity. We collected TFH and TH cells. We sequenced the transcriptome, the genes that are active in all these cells, and we compared the transcriptome of these cells. We can discriminate a signature that can discriminate the cells that are type 1 and the cells that are induced with type 2, and we can discriminate TFH, the big balls, from TH, that are the small balls. We can see what are the genes that are different between TFH cells and TH cells. That is easy. This is just to confirm that we can pick up the right signature genes as represented here, but the key problem that we did not know is whether and how we can discriminate TFH1 from TFH2. We did this with a machine learning approach using computational methods to try to find out what is the minimum signature that can discriminate TFH1 from TFH2 cells. To validate this, we used data sets from different laboratories obtained with helminth infection and LCMV infection, a type 2 and a type 1 response. And our approach can accurately classify, so we could derive a signature of genes that could classify the different adjuvants that we used, but also the public libraries from LCMV and helminth infection. And as you can see here, there is no difference between the test data sets and the training data sets. Finally, we use single-cell RNA sequencing to sequence the transcriptome of all the individual TFH cells that are involved in this type of responses. We isolated from the full transcriptome of the T cells the ones that are TFH, and among these TFH, we found that in animals that receive a type 2 immunization, we have the majority of TFH cells with this type 2 signature, with a few that are of the heterologous type, and vice versa. Probably also explaining why when we have an immunization with this type, we always have a small proportion of antibody of the discordant isotype that is also produced with this immunization. So we believe that we have now confidence that that this classification between TFH1 and TFH2 cells can be established, and we found the signature transcriptional profile that can allow us to discriminate TFH1 from TFH2 cells. And the very last slide to show the computational person that did all this computational work, Soumya Kumar, and some people that were involved in this study, Filipa and Rodrigo. Thank you.
Okay, well, a while ago I was very unpolite, I realize now. I did not introduce properly Professor Miguel Prudencia. I'm going to do it now. And in a small, short phrase, you can say that Miguel is Mr. Malaria Man in Portugal, and you all know that. And now we are going to have Professor Thomas Enscheid that's going to talk us about diagnosis, and this is almost aim in life, and he knows it all about. Thank you very much, Thomas.
Well, good morning to everybody. Thank you for having me. It's just going to be a fairly quick presentation about some aspects of diagnosis. In fact, I was thinking a bit about what to talk about, and I thought, thinking about the title of the conference and the challenge for the future, I really had to show, the quote from Niels Bohr, very difficult to predict the future, well, any prediction, especially the future. What I wanted to address is, as a matter of fact, a bit like what are diagnostic tests, what are in principle the questions we have to address if we want to get better diagnostic tests. As a matter of fact, we are now after prevention and prophylaxis and we come to disease. Obviously what you want is you want to establish what kind of disease it is to be able to treat it properly. So we want a diagnostic test, a laboratory diagnostic test, at least to confirm our suspicion. And most tests which exist, which are quite a lot of them actually quite old, have limitations. And they are this would be the wish list of those things which I would like to improve in tests, apart from what everybody knows, specificity, sensitivity. That means better tests detecting better with less false positive and less false negatives. But there are quite a lot of other aspects which could be considered when you think about diagnostic tests. Obviously, the list is quite extensive. I don't want to talk about all of it. Certainly, one aspect which is very important, especially now at the times of COVID, is that diagnostic tests which are used to diagnose a disease have different characteristics than when you use them for screening. As a matter of fact, they are very, very different in terms of what challenges they pose, in terms of what they should do, how easy they should be, what kind of sensitivity and specificity they should have. But apart from this, of course, we also want to think a bit globally. That means you should not only, and that's not only a question of globally, as a matter of fact, in the COVID pandemic, we can see it. PCR tests, which are mainly used, are very expensive tests and cannot be used in all circumstances. So we want tests which are simple, easy, cheap, which can be used at the point of care, or at the moment, actually, at the point of a suspected case. So they have to be really that easy to be able to use them, and quite cheap. So there is a question around what can we do to get tests that don't work only in our laboratories, but in resource-poor settings. And I just wanted to mention there's actually, since 2008, there was a Congress in Maputo. There's a declaration called the Maputo Declaration, which actually looks into this and tries to provide, certainly in the case of Africa, access to laboratory tests. And on the other side, I also just want to mention, I don't have time to go into this, the question of patents. Most people think that patents, that is something inherently good, has to exist for anything that we investigate and we develop. And I found it quite interesting. This is from The Economist, you know, the newspaper or the journal that defends obviously the economy, and they are actually very hostile to patents. And I think it's very interesting to read this paper and the reasons why they oppose patents and why they think that's an obstacle to progress and not an advantage. It may be an advantage to earning money, but it's not an advantage to progress. From this, I hope if we manage, I want to show you a short part of a video before we go into some details.
Elizabeth Holmes promised the world a medical revolution. At 19, she claimed to have invented a miniaturised machine that, with a single drop of blood, could map our health. Her boast was serious diseases like cancer would be prevented before they happened. And it was not only a wow moment for patients. High-profile investors poured a fortune into Elizabeth Holmes' company, Theranos. Their money guaranteed she became Silicon Valley's first self-made female billionaire. But her invention was an invention. It didn't work.
So this is just to show you one of the high-profile cases, if you don't know it. This woman actually claimed to have developed a test that on a blood drop could diagnose pretty much everything. I think at one stage she was valued of being worth something like $10 billion. And as a matter of fact, she's in court now. It's one of the biggest fraud cases in the United States. I think this is very, very interesting to read this, or to study this a bit and to read about this. A very interesting book about this. Because it shows that quite a lot of the hype around this new invention, especially in diagnostics, do not necessarily lead to some really practical or good practical outcomes. With this, I just want to say a bit about what I'm actually doing. I'm actually working a bit more on practical things, with my background as laboratory trained physician on diagnostics. As you can see, a couple of our papers I've been working on different stains, especially fluorescent stains for TB diagnosis. As a matter of fact, we did not only try to improve them, but one of the things we found out just by chance, like good science usually works, it's not the eureka moment, we just found out by chance is that this stain, which was only meant to stain mycobacteria, also stained schistosoma eggs, which has never been described. We've been working on this with some, as you can see, very nice images here at the bottom. Another aspect we have been working on is to improve histology diagnostics, also using the same stain. As a matter of fact, that might have implications for a lot of other people. In histology sections have to be embedded in paraffin to be cut, and when the paraffin is taken, you destroy pretty much everything in the membrane that's lipidic, and that also destroys bacteria if they're present. And as you can see, we just had the idea to use a hairdryer and blow the paraffin away, and as you can see in the results, we got excellent results with our new method as compared with the usually xylene deparaffinization method. So that would be a huge step, simple step forward, and in my opinion represents actually quite a lot of what my philosophy is, which I would describe as Occam's razor. Occam's razor is on the left, which most of you know is this monk that did describe this philosophy that if you have complicated explanations, simple explanations, the simple explanation is correct. This can be applied, in my opinion, also if you have complicated solutions and simple solutions, you should use the simple solutions, which is also represented on the right. The motorsport, mainly from Germany, has been translated into English. If you think about rally cars and racing, they're completely stripped of anything unnecessary, with the idea that if it's not there, it cannot break. So I think that's what we have been trying to do in terms of work. And I can show you, we have not always been very successful. I've been working also on a sensitivity test for malaria using hemozoin, one of the byproducts of the parasite, which is reagent-free detection, very, very quick. We did actually develop the quickest phenotypic test in the world. After 12, 16 hours, we can detect the sensitivity of the parasite, but the methodology is so complex that trying to implement this in Africa, we actually did it. We took it to Gabon and managed to make it work, but it's actually not very, very easy to do. So I don't believe that it would ever be really useful for a real-world alternative for sensitivity testing. With this, I just wanted to say that in the world of, coming back a bit to what I said in the beginning, in the world of diagnostics or research, I think we're actually not sort of like having a problem that we have too little information or too little research. You might have too little correct or good research, but especially in diagnostics, what I find is we have lots of research on diagnostics. We have lots of diagnostic methods. These were just the first couple of papers I printed just as I found them after a search. And to me, as a matter of fact, it looks a bit like it's a solution looking for a problem, because there are too many methods, and I would say 99 of these 100 methods are never going to make it into real life. Also not why, because again, and this has to do a bit with my philosophy, rapid diagnostic tests, not only now like in COVID, but in malaria are the ones, which is publication from 21, that confirms that after 2010, when they made it into real life, they have made a huge difference for diagnosis in Africa. And this is indeed like a very nice way to show how a diagnostic test, one diagnostic idea, can really make a big difference. With this, just quickly about these tests, although they are so nice, we all use them at the moment to diagnose or to screen, more to screen than to diagnose COVID-19 or people infected with it. And as you can see, this is, in my opinion, a bit nonsensical. If you look at this is from the DIS, from the DGS. They say that these tests have to have a performance of at least 90% sensitivity, more or less, or higher. This is nonsense. The studies that are coming out now are showing that these tests at best have an overall sensitivity, Cochrane review of 50%. The study on the left is a real world in 1,500 people, and it shows that asymptomatic people, it has only a sensitivity of 44%. That means the idea that these antigenic tests perform so well that they can be used easily and very successfully in screening is at best hopeful, I would say. It's obviously the best we have. I don't want to argue against these tests, but the idea that these tests are really a true solution to contain the pandemic, I think I would side with quite a lot of other people who are already saying that this is a rather doubtful exercise. And it shows once again, we do not have a simple test for screening unless we would do PCR, which is impossible, or we use PCR and we do pooling, but even that is impossible. With this PCR, molecular methods are always obviously now known to everybody. Everybody talks about it. This is the big advance. Certainly it is. It detects the lowest number of any organisms, but also has limitations. In the case of COVID, it's very easy to understand. We're having the problematic that we have false positives for a rather long time after the disease. So I think that shows again that a test that is very, very good for a certain situation, which might be good for screening and diagnosis while people are infected or acutely sick, cannot be used for the weeks afterwards. So there is no single test yet that really solves this, and I think COVID does actually show this quite nicely. It may it be the antigen test, may be the PCR test. In fact, the PCR tests, which are very much hailed as the big breakthrough in the last 2 decades and so on infectious diseases, have actually quite a lot of limitations. This is just from a paper, from a review on sepsis diagnosis, one of the diseases or disease syndromes, which has a very, very high mortality and where we need to identify the bacteria very quickly. With culture method, traditional method, that takes very, very long. So this is just comparing the methods which exist. And when you look at that, I think it looks very impressive because whatever the method, forget what says here, it's just to show you what you have and that it takes a couple of hours only to perform these methods, they can detect hundreds of bacteria and viruses. So we should actually all jump and be happy and think this is the solution. As a matter of fact, it's not so easy. If you look at the clinical impact that has, how much that does actually change clinical management, these tests are actually not performing so well. This is actually even the latest test, which is the so-called carriers test, where you do sequence the organisms. You just take a bit of blood, and then 1 or 2 days later, you get the organism, and it has been used quite a bit in the United States for identification of organisms causing infections. Costs like almost $2,000 or something in the order, so it's very, very expensive, and they looked at the impact. And in this paper, they come to the conclusion, as you can see, that the impact is rather very limited. That means in terms of what those tests do, apart from, like remember, I showed you all these diagnostic tests for malaria, which might or might not perform very well, is also another question. How really useful are they? So with this, I just wanted to show you, also to show a picture of me in Africa, installing it, of course, but mainly to show you that there is one exception to that. That means it's not really an exception, but it's a disease which is TB, tuberculosis, with a very high lethality, case fatality rate of almost 2/3 untreated. And this in Africa is difficult to diagnose because they only use microscopy, they cannot have culture, and there's a gene expert, a method which is being supported and funded mainly by Bill and Melinda Gates Foundation. The equipment, which costs normally $100,000 and the test $100, is available in Africa for around $10,000 and the test for around $8 or $9, but even that is very expensive. But this is actually a huge advantage because it detects resistance. So this is a very severe disease in which this may be a big advance. So with this, I just wanted to actually already finish this talk, talk a bit about all these different methods, just to say that the last area we're obviously talking about is biomarkers. Biomarkers has been quite a sexy subject, actually even in the past. That actually from 2000 onwards, people have been saying we do not want to identify the organisms to make the diagnosis, but we eventually want to see in a patient if he has a certain pattern of a biomarker which would allow to diagnose that this person has disease A, B, or C. And there has been a huge search for biomarkers, and I have to say, if you look at this, I am not so optimistic that somebody will find a very, very useful biomarker. The most useful one has been procalcitonin, which is a biomarker that allows distinction more or less between viral and bacterial infections. But as you can see, I just showed this here I found this on the right. These are obviously EU-funded projects with lots of money. This is from 2010. And I think you and me, we don't remember any big breakthrough in some new biomarker. So I'm not saying that they did bad research, but they certainly did not find the biomarker which would change that. Having said this, there's obviously a lot now going into microRNAs or non-coding RNAs as new biomarkers, which again I think is a very interesting, sexy subject. I still have to see how useful they might be in clinical practice. And with this, I just want to finish and say obviously that in the end, one of the areas which of course we also will still see quite a lot of developments is antibiotic resistance and detection of antibiotic resistance, which is not culture-based. Obviously that's also all going to be again molecular-based. But if you think about this, any molecular-based method can, by definition, not be a sensitivity method. That's illogical, and most people don't understand that, because molecular methods cannot detect if an organism is sensitive. Molecular methods can only detect resistance genes. That means a molecular method, by definition, is a resistance test and not a sensitivity test. And because I have to infer the sensitivity from the resistance, there are some tricky details in practice, especially with certain bacteria. So with this, I just wanted to finish. I'm probably already talking too much. And I found this quote yesterday, and I thought that's actually quite funny. It certainly applies to diagnostics. Okay, thank you very much.
Okay, so we're now going to the last talk of this session by Professora Maria Emília Valadas, infectiologist at Hospital Santa Maria and also a professor at Faculdade de Medicina, who kindly agreed to give a closing talk for this session. Although it was not originally planned, she was very nicely accepted the challenge to give a small talk about treatment and management in infectious diseases, which we are now going to hear. Thank you.
Thank you very much, Miguel. Well, like Professor Miguel Prudencio said, I'm like the buffer on this session. And I'm trying to leave like 5 minutes or so to have some questions. We are few, but I'm sure that we are going to have few questions, but clever questions. Well, they were talking about vaccines and diagnosis and immunology, difficult things. I'm going to talk about what interests me more, that's to treat patients. And in this respect, and related to COVID, that's the subject at the moment, there are some news, good news, but not so good as we wanted. This is just to remind us that vaccines are excellent. I hope that everybody is vaccinated. I really hope so. But what we also need is effective treatment, because some people, for whatever reason, they are not going to be vaccinated and they are going to be sick. We had some treatments or some promises of treatment over the last month, and everybody heard about chloroquine and ivermectin. They did not work. Then we had another famous treatment, famous because it was shown in a press conference of an ex-president, and unfortunately also did not work. But this remdesivir was used only for very sick patients. Did not work for many reasons. We don't have time to discuss that. And then we had treatments with monoclonal antibodies that were also very promising, but they have to be given in a certain way, and it's not applied to many places where the patients are. So nothing of this did work. And of course we had also the old corticosteroids that always work in very severe patients, whatever the pneumonia is caused. But we don't want to reach that stage. That means corticosteroids always work and work in COVID, but we would prefer not to have these very sick people. So we did not have very much until now, until very recently. And these are two drugs, a drug and a combination of drugs from Merck and from Pfizer that are very promising. The molnupiravir and the Paxlovid, I'm sorry, I did not learn very well to say that, are very promising, and it's a 5-day treatment with pills, not intravenous. And the first results, they look very promising. Of course, that Pfizer and Merck have promised, if FDA approves very quickly this treatment to treat the patients, they are going, one is going to, well, they both are going to help the world to construct very quickly the drug to be available to more and more patients. It's really a big advantage over the last months, and we are very happy with that. I have to say that, and I don't want to talk for a very long time, but I have to say that I feel very lucky on this, because in the 80s I lived the other pandemic, the HIV one, in a time that we did not have drugs to treat the patients, and 99% or more than 90% they did die. And then we start to have combination of drugs, and things did improve, and on the end of 90s we started to have a very good cocktail of drugs, and now nobody dies of HIV unless he or she is very unlucky. And we are going to see exactly the same here. We start with 2 drugs that are promising. I'm sure that in the next few months we are going to have new drugs and new combinations, and this is going to be a good way to live with this virus. Just to remind us that the antiretrovirals to treat HIV, they took possibly 17 years to be available for most of the people. And now we have drugs that are available after, 18 months or 1 year and a half, which is really amazing. The same with the vaccine. So I'm not going into details, but these 2 drugs here, or the the one drug and one combination, because one of them is a combination with ritonavir that is also used to treat HIV. Again, we are trying to reuse drugs that are already known for the areas. And the good thing about this is that the risk of hospitalization and of severe disease, it's much, much lower, and we are just at the beginning. And I'm very, very optimistic about these treatments. The same here. And just to remind us that 1 year ago, we had the news about the very effective vaccines that fortunately they are very effective. And 1 year after, we have the good news about a very effective treatment. So despite the fact that the new variants are around and everybody's a bit concerned, I'm very optimistic about what we have now. Thank you very much.
Thank you very much, Emilia. It was very nice. Thank you all for your excellent talks. And I'd like to ask the audience if they have any questions, please. Raquel, okay.
So I have a question for Thomas. So you were saying, I might say something wrong, so correct me if I say something wrong, but you were saying that there were a lot of diagnostic tests for available at the moment. But don't you think that there's still room for improvement in that area? Because I might be wrong, but in the dry season, you have sub-detectable levels of gametocytes in the patient's blood, and I know that they're not very easy to detect, right? So don't you think that there's still room to improve there? And that I know there's also this problem in Africa where the tests are invasive in the sense that you have to draw blood out of patients, and I know there is a test that is being developed on the diagnostics with saliva. So don't you think also that could be the way, you know, to do something that is less invasive and also, of course, highly sensitive?
Well, thank you for the question. Well, of course, there's always a way to improve things. So there's no doubt about this, and I did not want to have my talks understood that there's no way. I have to say, like, to answer that question, I think there are two aspects which I think are important. The first is, it should be quite clear, and I took that actually from my presentation, is because I thought it was inherently clear that if you talk about diagnostics, diagnostic is in this sense, or at least in my head, something that you use medically. That means you use it in the context of a patient. That means it influences what you do to the person. Now, the scenario that you described, for example, gametocyte detection, does not have any, at the moment at least, palpable or immediate consequences. So what you would, I think, or what you should call that, I've been talking about this a lot, would not be diagnostics, but would be detection methods. So I think if you divide the world of, let's say, not diagnostics, but of methods, okay? You can divide them in detection methods and diagnostic methods. In the world of detection methods, for many, many reasons, epidemiologically or whatever, of course you can, and you do have lots of advances, okay, which can be very useful for other purposes, okay? So yes, of course there can be and there will be lots of things happening. I do remember I wrote a paper some time ago which I called, it was about PCR in 2003. We actually wanted to call it Much Ado About Nothing. Why? Because we were reviewing 25 papers on PCR diagnosis of malaria, and most of these papers, people collected blood in Africa, took it back to their countries of origin, for example, Canada, and 3 months later diagnosed malaria, and dared to write in the paper, quick diagnosis of malaria, because in the lab it took them 20 minutes. So that does not make sense. But of course, there are methods that could be improved. Now, of course, non-invasive methods can definitely change everything. I took one slide, which was the second video, which I wanted to show, which I'd like to show. Not that I'm a big friend of Star Trek, but if you do remember Star Trek and Mr. McCoy and Dr. Spock, they have the ultimate instrument which we would like for diagnostics, which is called the tricorder. You know, this instrument that looks like a shaver, so zzz, which diagnoses everything, and the most important is not only very sensitive, specific, above all, it's non-invasive. So yes, anything that goes away from blood and reduces even risk, certainly very, very good.
Any other questions?
I have a question for Thomas as well. Regarding the tests that are not very sensitive, the antigen tests, one thing that this pandemic also taught us is that maybe an imprecise test that is rolled out is better than no test at all. There are some mathematical models suggesting that frequent testing, even with these not so precise tests, have a good outcome. And the practical results, I think that I saw a study with twice weekly testing of students in the US colleges, self-tests, that were very effective in preventing outbreaks, and probably it would not be possible to have such testing scheme with tests that would be more reliable, but maybe could not be implemented. So what is your view on that?
Well, I think, as I said, as I tried to say, thank you for the question. As I said, and tried to say during the talk, any test is better than no test. That means if you have a test, you have no test, you have 0% sensitivity. Anything is worse. I have to say, I doubt a bit these figures because even the studies are rather a bit older. Now, what they have shown is, but this was kind of fairly artificial and also in England, that if you do a testing scheme actually on a daily basis with the best tests, of course, that you can pick up quite a lot of people who might become infected and go on to transmit, and you reduce transmission overall. Now, you also have to know that this is all also done, or most of this done with variants from the past, and does not apply necessarily to Delta, which is much more infectious, and has one aspect which is important, that the period during which it is, as a matter of fact, transmissible is much longer, even before any symptoms. So, well, yes, in certain circumstances, you can create schemes where, let's say, even if you have a bad test, it has some utility, and they have some utility. But I have to say, the scheme, for example, of this American school, I have some doubts about it, that 2 weekly testing is really good enough, because there are other studies that show that you might still have quite a lot of people that might pass through the gap. The main aspect here is, and it's an important aspect which most people forget, is that the sensitivity and specificity is actually quite secondary. What most people forget is that you have to know how to implement this in terms of the underlying prevalence. That means if you have a test that's even 95% good, but you have a very, very low prevalence of 0.5 or 1%, the number of people that you produce, the number of false negatives and false positive changes completely than if you have a 20% prevalence. And this influences a lot where you can use these tests, but probably that goes into too much detail.
Okay, we have time for one more question. Does anybody else want to ask a question? If not, I was going to ask one to Emilia, but you can ask one to me.
So I want to ask you in the results that the vaccine that was approved, the RTSS, it is a subunit vaccine. So regarding what you presented about the need to target different stages of the life cycle of the parasite, how does the effectiveness of this subunit vaccine compares with all organism vaccines, not only the one that you are developing, but with the radiated sporozoites,
Sure.
regarding these mechanisms?
Yeah, so the subunit, the RTSS vaccine, the subunit vaccine that is now endorsed by the WHO, has, like I mentioned, about 30% efficacy in preventing serious disease, whereas whole organism vaccines, and I'm not talking about the one we're working on, but irradiated sporozoites and others, they have actually shown up to 100% sterile immunity in clinical trials. So in general, the data support the notion that whole sporozoite vaccines are much more effective than subunit vaccines. Now, what people are also trying to do is to improve subunit vaccines and combine different subunits. So RTSS is based on a single antigen, and there's been attempts to combine multiple antigens. But for now, what we have at the moment is that sterile immunity is achieved with whole sporozoite vaccines better than in any other option. Did I answer your question? Okay. And final question. Anybody? A question? If not, I just want to ask one question to Emilia, which is about what you said about treatments. So I've been following as closely as possible the progress on vaccines, not so much on treatments for COVID, but I was under the impression that monoclonal antibodies were also going to play a critical role in this, and you mentioned them briefly, but the two, just to clarify, the two pills that you mentioned, they are not monoclonal, they are antiviral, not monoclonal antibodies. And how do they compare in terms of effectiveness against disease with monoclonal antibodies?
Well, Miguel, the problem is that most of the people that are going to be infected don't live in Europe. And monoclonal antibodies are expensive and difficult to administer to the patients. So this is a pill, 5 days, only 5 days, oral, and this is completely different, and it's possible. And I'm very confident on that. Luis is going to say something about monoclonal antibodies? Yeah.
Yes, just a follow-up. All the clinical trials of these were done with placebo, non-vaccinated people. Yeah. So in a situation where we know that the vaccines prevent against serious disease, and where most people in countries that are as spectacular as ours in vaccination are almost all vaccinated, the real question is, how does these drugs work with vaccinated people?
Yeah, that's true. But I think we are just at the beginning, and I hope that the people that are here listen to us take home also a positive message. This pandemic has many negative things, but one of the positive things is that in terms of techniques, in terms of vaccines, in terms of development of drugs, we are living a very happy time, I think. Then we should look a bit more to the positive things and not so much to the negative ones. And I think a very good time is coming, and the COVID and everything that we are going to learn about COVID and treatment, vaccines and so on is going to be used, well used in other diseases, in other infections. Then enjoy also this time, please. Okay, thank you very much to everybody. Thank you for, we are on time, just 4 and a bit minutes late, but thank you very much. Thank you for all the questions, and thank you to all, and I think it was very productive.
Thank you.
Thank you very much.